Yes.
Okay, please respond in the class.
Yes.
Okay, so when we talk about the chapter Heredity and Evolution, let us go through all of it once. So, first we should know about what genetics is. So, genetics is that branch of biology which will be dealing with the study of inheritance and variations. It tells us how inheritance happens and how variations take place. Now, what is inheritance? So, when the characters are transmitted from the parents to the offsprings, then we call it as inheritance, meaning the characters are being inherited by the offsprings. So, it travels from one generation to the next generation. Then there is variation. Now, variation, it is the degree, like it is the difference of characters in the offspring, meaning how they differ from their parents or some new characters that are introduced into them. So we call it as variation. So this is like the degree from which the parents and the offspring differ from each other. And it is also the introduction of new characters. New characters are being introduced in this. Now, when we talk about genetics, so the whole entire thing that will cause the genetics to happen will be obviously the DNA. And that functional part of the DNA is called as the gene. So everywhere, the genes, they govern the process of inheritance. Is it clear, students?
Yes, sir.
Now talking about the difference between genotype and phenotype. Now genotype, it's the way the genes are situated, whereas the phenotype, it is the observable characters that are expressed of a genotype. Can anyone give me the example of a genotype and phenotype, students? Yes, give me one example of a genotype and phenotype.
I don't know.
No idea.
No.
Abdul, what about you?
No.
So you people are not studying for your exam?
Studying.
See, genotype, if we say that the height is tall or the height is dwarf. So tall and dwarf are the things that you can observe, you can see and you can make out, okay, that something is tall and something is dwarf. So that is the phenotype. Whereas what represents this height will be the genotype. For example, capital T is the genotype of tall and small t is the genotype of dwarf. Same way, there are multiple genotype and phenotype. Now talking about genetics, so we must know the father of genetics, so that is Mendel. Gregor Johann Mendel, he gave the laws of inheritance. He said that, okay, inheritance takes place and how does it take place is what he explained by performing the hybridization experiment. So he performed an hybridization experiment on the pea plant. The scientific name of pea plant is Pisum sativa. Now the question arises that why did he choose the pea plant? Why not any other plant? Can anyone answer that?
Can you repeat what you said?
Why did Mendel choose the pea plant? Why not any other plant?
Yeah.
See, the reason why Mendel chose the pea plant is because the pea plants, they have,
Because one for the traits, ma'am. Like they're easy to breed also.
yeah, they show 7 pairs of contrasting characters.
Yeah.
So in total, 14 characters are represented by them and they are bisexual in nature, meaning they'll do self-pollination. So no pollinators externally are required for this process. Apart from that,
Yes.
they make many offsprings. So as a result of their reproduction, several offsprings are there. That means several different kinds of readings will be there. Now, when we
Yes.
talk about the 7 pairs of contrasting characters that were actually taken into consideration, so it will be the stem length, pod shape, seed color, flower position, flower color, and pod color. So these are the different types or the 7 types that we discussed that, okay, these are the 7 pairs of contrasting characters that were taken into consideration by Mendel. Now, in all of these characters, there are 2 alleles, that is a dominant and a recessive. So these are the contrasting pairs of characters. So always remember that one gene, it has 2 alleles present. And what is an allele? Allele is like a pair of contrasting characters. For example, if we talk about the height of the plant, so tall and dwarf, these are the 2 possibilities. One is the dominant and the other is the recessive. Also, tall and dwarf, they are the alleles of each other. Clear, students?
Yes.
Now, with respect to the genotype, there are 2 conditions that must be taken into consideration, that is homozygous and heterozygous. Now, homozygous condition is the one in which the same kinds of genes or the genotype is there. For example, if capital B is there and small b is there. So the capital B, it represents homozygous dominant. Homo means same. And small b represents homozygous recessive. The other condition is heterozygous condition. Heterozygous, hetero means different. So when capital B is present with small b, so that is called as heterozygous condition. Now tell me one thing, students. When a dominant trait is present along with a recessive one, which one will get expressed? The dominant or the recessive?
And the dominant one.
Correct. Always. It doesn't matter in which type of cross that you are talking about. The dominant trait or the dominant gene will always express and the recessive one will always express in the homozygous recessive condition and not in any other condition.
Yes.
Now, after doing the experiment of hybridization, Mendel gave the laws of inheritance. Now, the first law of inheritance is the law of dominance. Now, according to this particular law, that the dominant trait, whichever is the dominant trait, that will be inherited by the offspring. Whereas the traits which are recessive, they will get suppressed. So this is the first law, that is the law of dominance. Then after that comes the law of segregation. So according to this law of segregation, while the gametes are being produced, the genes that are there, so 2 copies of each gene, they also separate so that one gamete can get one type of gene and the other gamete will get another. So that when 2 gametes will be fusing together, then the combining from each parent will be there. So that is the law of segregation. And lastly, the third law is the law of independent assortment. And according to this law of independent assortment, whenever, mostly in the case of a dihybrid cross, wherein you're taking 2 characters into study. So in them, what happens? That the pair of traits, for example, if there is round in yellow, wrinkled in green, so the pair of traits will express themselves separately or independently, and it will not depend on any other trait, meaning there will be no blending, there will be no mixing. It will just happen flawless. Clear, students?
Yes, ma'am.
All right. Now, moving forward. Now see, in this, Mendel, he gave the 2 crosses, monohybrid cross and dihybrid cross. Both of the crosses we will be studying. But before that, you should know as a rule. Okay, learn it as a thumb rule that the phenotypic ratio of the monohybrid cross in the F2 generation, meaning the second generation, will be 3:1. And the same thing of a dihybrid cross will be 9:3:3:1. Always remember the phenotypic ratio is taken out for the second generation. So these are the two types of phenotypes that are observed in the case of monohybrid and dihybrid crosses. Any doubts, students?
No, no.
Okay. Now after that, we will be talking about the monohybrid cross. Now, monohybrid cross is that cross wherein only one character is taken into consideration. But before that, you should know what dominant and what recessive traits are. Now, dominant traits, these are the ones that express themselves in an organism. That will be shown, that will be expressed, we'll call it as dominant trait. And the trait that gets masked, meaning that will not be able to express itself whenever a dominant trait is present. So we'll call that kind of a trait as recessive trait. Meaning in the presence of a dominant gene, it will not be able to express itself. And it will only express when the recessive gene is absent. Like whenever the recessive gene is present in a homozygous recessive form, then only it can be expressed. So these are the two traits that I have talked to you about. Now, moving forward, talking about the monohybrid cross. Now, when only one character is studied, meaning you are studying how the inheritance of a single character is taking place, so we'll call it as a monohybrid cross. Okay, monohybrid, mono means one. So we study this cross, and with this we come to the, basically we know that during this process only one character is studied. So now to make the crosses, let's just say we are crossing the height capital T, that is tall, with small t that is short. So in the F1 generation, if you see here, the crossing has been done like this. So capital T gets crossed with the small t and then again with the small t. So in the first generation, all of the offsprings, they are heterozygous tall because capital T is present. So they will not be able to express the recessive character and they will only express the dominant one. Now, to find out the F2 generation, meaning the second filial generation. So what do? We do selfing, meaning you take any two of the F1 offsprings and cross them together. So when we cross them together, so what are the possibilities in the second generation? We get capital T, we get capital T, small t, then we get capital T, small t again, and small t, small. So all in all, if you see here, 3 of them are tall and 1 of them is dwarf. And if you remember at the end of the like the introductory part, I said that the phenotypic ratio, meaning the ratio of the external observable characters, it will be 3:1 in the case of monohybrid cross. And that's what's happening. In the monohybrid cross, 3:1 is the ratio that comes out to be. Any doubts up until now, students?
No.
Okay. Now moving forward, the next cross that we are going to discuss will be the dihybrid cross. Now dihybrid cross is when you're studying the two characters. That's how you get a dihybrid cross. Now for a dihybrid cross, for example, you're taking the height of the plant and the color of the flower, or you're taking the seed shape and the seed color. So depending upon that, Like for example, the seed color you're taking, round and yellow, that is the first one, which is basically the dominant. And the recessive one is green and wrinkled. So when you cross them together like this, see here, round and yellow crossed with green and wrinkled. So what do you get after that? You get capital R, capital R. From that you get the gamete, that is capital R and small, like capital R, capital Y. And the next one you get small r, small y. So that is how you get the information for the first generation. Now, for the second generation, you have to take out the gametes yet again. So you have capital R, capital Y, capital R, small r, capital Y, and small r, small y. So now you make a Punnett square like this. And in the Punnett square, you make crosses. So this is a Punnett square wherein the set, like the 4 gametes that you have received, you put one on the top and one on the left margin, and then you cross them together and note down their genotypes. Now in that, what will you observe? You will observe that this particular cross has different kinds of gametes and different kinds of phenotypes. And at the end of this, when you find out the dihybrid ratio, then that comes out to be 9:3 is to 3:1. This is the phenotypic ratio of a dihybrid cross. Is it clear, students?
Yes.
All right. So
Yes.
now, this was it. Now, students, this was one question that we have also done in the class. So now you people try to, first I'll teach you sex determination. Now see, sex determination is the procedure. It is the process in which the sex of an individual is determined depending upon which kind of genetic material or the chromosome composition is there. So in the case of humans, if we think about it, so the males in the human beings, they are like their genotype is capital Y and like X and Y. Whereas in the case of female, it is double X's. So when the gametes and crosses are being done, so we'll make it like that. So male will be XY. So it can either donate the X chromosome or the Y chromosome. Whereas when we talk about the female, so it like in both the cases, she'll be donating the X chromosome. So when the X of the male combines with the X of the female, so we get XX, that is a female child. When the X of the male combines with the another X of female, then we get double X's, that is again a female child. But when the Y of the male combines with the X of the female, so at that time we get the XY, that is a male child. And when again the Y of the male combines with the X of the female, we're going to get XY. So this is the genotype, and we can say that in this type of cross, meaning when like whether to decide the baby will be a male or a female, so that is a 50-50% chance. It can either be a male or a female. Now, a very common question would come along that who decides the sex of the baby, or why is the father responsible for deciding the sex of the baby? So what would you say, students? Tell me, what would be the answer for it? Who decides the sex of the baby, the mother or the father?
I'm the mother.
Hmm? It'll be the father. Why? Because the father is a heterozygous condition. X and Y is then in the father. That is why. So with this, the chapter has been done. But there is this one question. So students, try to attempt this question and tell me what would the answers be. Try it in your notebooks. Yes, very good, Aisha. Your answer is correct. What about others? Galvin and Abdul, did you get the answer? Yes or no?
No.
Hmm?
No.
What's the problem? Tell me. Why aren't you saying anything? Ask me. What's the problem? Which question were you having a problem in? Tell me, which question do you have a problem in? Am
Yeah.
I audible?
First one.
Yes. Yes, tell me, Abdul.
Yes. First one.
First one, see, you make a cross. You take capital B, and then you cross it with small b, small b. So, capital B and small b, that will be the genotype for all the offsprings of the first generation. And capital B, small b together, it will represent the dominant factor only. That will be the blue color. Clear?
Yes.
Yeah, so that is how it will be done. The second part will be what percentage. So then you have to find out the F2 generation. Now, when you find out the F2 generation, so in that you will be seeing how the organism, like this thing, you take two offsprings of the same this thing genotype and you cross them together. That is capital B small b crossing with capital B small b yet again. And that is how you're going to get the particular genotypes of each of the organisms. And in that you'll see, you'll find only one small b small b. So 1 upon the total number of offsprings, that is 4 into 100 to find out the percentage. That is how you get 25%. And the phenotypic ratio will naturally be 3 is to 1 in the case of a monohybrid cross. Now students, solve these MCQs so as to practice what you have learned. Tell me once you're done. So you can write these in your answer, like the notebook, and practice.
Done, ma'am.
Yes, all of you are done.
Yes, ma'am. Done.
Finish.